In this video, we're going to continue to look at what's going on with DNA inside of cells and consider what changes in the DNA can have effects on both transcription and translation. And really, when we look at this process of mutations, we have to think about that idea of genes code for functional products, and mutations in those genes are going to change those functional products that are coming out of there. And when we go back and look at this idea of mutations, we have to remember that this is something that really is what drives evolution, is these changes in the existing genes, and the products that are made are going to go through the process of natural selection, so that organisms that have something that allows them to survive better in the environment are going to be able to reproduce more. And so the ones that develop mutations that are beneficial, that could potentially allow them to reproduce more, are the ones that are going to come to dominate that population. And that's where we get that idea of Darwinian evolution by means of natural selection. So if we take a look at some of these effects of mutations, we can take a look at a single type of mutation that occurs known as a point mutation. And here you're looking at a change to a single base in that DNA. And if we look at this example depicted over here, it shows you what's known as a silent mutation. And this is silent because there is a change in the genotype, so what is actually possible from the gene, as opposed to a change in the phenotype or the product of the gene here. And when you have a silent mutation, in this example we have this DNA region that is coding for this message right here, and then that is coding for this order of amino acids that would be inserted as the ribosome is reading the message right here, and the tRNAs carrying those amino acids are coming in and dropping off those amino acids for the protein. Well, in this example here, there is a single mutation where this codon changes in the DNA from a GTA to a GTT. And if you think about it, when that piece of DNA is read by the RNA polymerase to make the complementary message, that is going to result in a single point mutation change to the message right there. And in this case, because we're looking at a codon that is degenerate, meaning there is more than one codon that codes for the same amino acids, well, here we have a valine at the beginning that is going to result in a valine being inserted into the protein. And so this mutation, while it did occur, is silent because you do not see an actual change in the functional product of the protein. But remember, all these different models for regulation of genes that can take place at the RNA level, that could potentially have a change on the folding of the message that could change whether or not it is translated. So here we're looking at it just in terms of it's silent in terms of the protein that is actually coded for by that gene, or by the amino acid that is coded for by that particular part of the gene. You can have a different type of point mutation out there where, again, you have a single base change. And here we have the CCC in the message is getting changed to an ACC. So there is one single base change. That first C is now changed to an A right there. And if we go over to our codon table and look at this, there is our original codon right there with the CCC, and that is being changed to this ACC, which codes for the amino acid threonine instead of the amino acid proline there. So here this is causing a missense. The ribosome is going to do exactly what is encoded in the message, because the ribosome doesn't read the message to make sure it's correct. The tRNAs are going to hydrogen bond with their respective codons that are out there, so they are literally just matching up the bases for those hydrogen bonds, and they don't know that there is a change in the message, much less a change that occurred on the chromosome level there. So this missense could potentially result in a change to your protein, which could affect its structure. It could affect the active site and what kind of substrates can bind in there. So a lot of different things could occur just from a single base change like this. And this kind of mutation is famous because this is what we see with sickle cell anemia, that there is a missense mutation that results in one single change to the amino acids there. If we move on and look at another type of point mutation, this one is called a nonsense mutation, because here your message has a codon right there that codes for an amino acid. In this case, UAC codes for a tyrosine. But over here we have this mutation that changed that codon to a UAG. So your C changed for a G right there. And this is a stop codon right there. So as your ribosome is reading along the message, normally it would be inserting an amino acid there. But here it comes along and it runs into the stop codon, which is that trigger for the polymerase or for the ribosome to grab that growing peptide chain and try and transfer it over to the amino acid that would be carried there, but there's release factors sitting in the A site for the ribosome. So the ribosome uses this as a signal along with other release factors that cause the ribosome to let go of that protein, and the two subunits fall off of your message and can get reused there. So in this case, it's nonsense because it is inserting a premature stop, and it is completely blocking the ribosome from reading anything after that period there. And so if you think about this, the closer you are with a nonsense mutation to the 5' end of your message, the shorter your protein is going to be. And you could potentially have a protein where you have a single amino acid inserted for your start codon, and then the next one could be your stop codon. So this would make something that is nonsense. One of the other kind of interesting things that we need to think about, the idea that the ribosome, all words are 3 letters long in the message. So the codons that are out there is what's being read by the ribosome to determine your order of amino acids. Well, what if you had a single base change that was an insertion or a deletion? And if you think about that idea, the ribosome is reading these words in sets of 3. Only now you've thrown an extra letter into there, or you've deleted an extra letter, and that's going to cause all of the amino acid codons that are there to be shifted over by 1, depending on if it's an insertion or a deletion. And if you think about that, now all of those amino acids are going to be off, and this could potentially result in the creation of a completely new protein that's out there. And again, when we think about this idea of mutations, it's not like organisms are mutating on purpose here. These are things that are just occurring as the normal process of DNA replication, that mistakes do creep in over time there. Not all of these mutations are going to be these small ones with single ones. We can also see larger changes that can occur. And this is an example where you can see what's called a chromosome translocation. And here you get a break in chromosome 4 and a break in chromosome 20, and the DNA strands are actually exchanged between these two chromosomes right here. And now you've got this mixture. So your chromosome 4 actually has part of chromosome 20 and vice versa. So there is this reciprocal change in your DNA right there. And if you think about if this mutation occurs in the middle of a gene, that could have some really weird effects, because now you could potentially be combining the effects of those two genes and getting a hybrid kind of protein. Again, that could be something that is beneficial to the cells, or if you consider, what if this is a gene that codes for a protein that's involved in cell cycle that tells the cell to go ahead and divide, and now you've moved it so the promoter is in front of it is one of those constitutive genes that is always turned on. Well, you think about that, in that scenario, now your cell is going to constantly be making a protein that tells the cell go ahead and divide, and that could potentially result in cancer, and that is what they do see with some of these chromosome translocations that are out there. Another one that is kind of similar to that when we look at these chromosome changes is what's known as an inversion. And this is a really interesting one, because what happens if you look at the order of these alleles on this chromosome right here, you actually get a loop that can form within your chromosome there, and that loop sort of folds over and breaks and then turns the DNA upside down and reattaches. And so here we've got this little change right there that has basically switched your A and B orientation right there. And again, think about that idea. If that's in a gene, that could totally change the protein. If it's in the promoter, that could totally change the RNA that is being expressed and when it's expressed, if we look at that idea of gene regulation. So these things do happen. There's really nothing we can do to stop them, because this is just kind of like your car breaking down. You don't want it to happen, but it does happen every once in a while there, usually when you're in a hurry to get somewhere. If we look at this idea of mutations and think about this DNA replication, they've been able to measure it, and they estimate that it's about 1 mutation for every billion bases that is copied. So when you think about that, that DNA polymerase that is involved in DNA replication is going to be a really good enzyme. And if we think about this idea, E. coli, it can replicate its chromosome. They have less than 1 billion. So you could have E. coli going through a cell division with no mutations occurring. Think about this idea for humans. We've got 3 billion bases per cell, and we're getting 1 mutation for every billion bases copied. So if you put the math together there, you're looking at 3 mutations are occurring on average every time your cells divide. But the power is that the enzyme that does this process, our DNA polymerase 3 that is out there, and to a lesser extent DNA Pol 1, they actually do have this proofreading ability where they can back up and scan the DNA that's been made, and if there is a mutation, then your width of your DNA strands are going to be either too skinny or too wide. Again, that idea of single and double ring bases on there comes into play. Your DNA should always be that same width apart, with 1 big and 1 small base that are complementing each other right there. So your DNA polymerase can fix your mutations that occur, but there are things out there that are known as mutagens that can result in an increase in the amount of mutations that are out there. And that can, again, affect our RNA and also our protein sequence there. And if we look at some of these chemicals that are out there, this is a normal kind of adenine nucleoside. Notice there's no phosphate, so this is not a nucleotide yet. But this one's kind of interesting because they've been able to chemically synthesize what's known as an analog of this nucleoside base right here. And instead of having an amino group hanging off here on this ring, they've actually got just a hydrogen. And that amino group is important because that's actually involved in doing the hydrogen bonding between the complementary bases that are out there. And what happens with this is, by chance, the DNA polymerase inserts this 2-aminopurine nucleoside with the phosphates added to it. This 2AP gets inserted into a new strand of DNA that's being made. And what happens is, where you normally would have an AT base pairing, after the DNA is replicated, this thing will actually cause a shift in the base pairing. So the next time the DNA is used, you're actually going to see it switch over, and the new daughter strands will have a GC base pair going in there. So every time this chromosome is replicated, the new daughter chromosomes are going to have that mutation as a result of this 2AP. We can also find nitrous acid. If you inhale this, this could get into your lungs, and it actually chemically modifies the bases in your DNA, and you get this deamination occurring in the presence of this nitrous acid. And this is kind of weird because you would actually form what looks like a uracil base, but the sugar coming off of this would be that deoxyribose. So this is deoxyuracil that would be in your DNA, and where we started out with a C, now we've got something that is going to base pair with your A. So again, a single point mutation could occur in the DNA as a result of breathing in nitrous acid, and that could potentially give you something like cancer in your lungs. If we look at this one, it's a really interesting example. Again, another completely synthesized base that's out there, but here, rather than the changes to the nucleotide base, the changes are actually occurring in your sugar ring. And so we've got our guanosine nucleoside over here with your 5-carbon sugar and your base hanging off there. That is your guanosine. But in this bottom one, we have what's called acycloguanosine. And if you look at this, it's missing the bottom half of the sugar, so there's no 3' hydroxyl group for the addition of the next base. If your DNA polymerase inserts this in, you're going to get what's known as a chain termination. Your DNA polymerase is going to stop right there because there's nowhere to add the next base. The last chemical that we can look at out there is what's known as an intercalating agent, and these guys will actually insert between the strands of the DNA, which changes the shape, and when the DNA is used, you're going to get your mutations. And the other kind of thing that we can see, instead of chemicals, we can have radiation causing mutations. Ionizing radiation will actually cause breaks in your DNA, whereas non-ionizing radiation like UV light results in the production of dimers that can change the DNA.